Welcome to Biotech Learning. In today's video, we're diving into one of the most essential tools in science, the microscope. What is a microscope? A microscope is an instrument that magnifies small objects, allowing us to see details invisible to the naked eye. It works by bending light or electrons to create an enlarged image of the object. History of microscopes. The story of the microscope starts in the late 16th century with Hans and Zacharias Janssen, who developed the first compound microscope. Then, in the 17th century, Antony van Leeuwenhoek, often called the father of microbiology, used microscopes to discover bacteria and protozoa. Principle of microscopes. When the sample is placed on the stage, the light from the light source passes through the glass slide after being reflected by the mirror. To provide a clear, magnified image for viewing the tiny details of the sample, the microscope's biconvex lens enlarges the image of the object. Next, the image is viewed from the other direction. Did you know there are two main types of microscopes? First, we have light microscopes, which depends on visible light to magnify objects. These include compound microscopes, bright field microscopes, dark field microscopes, phase contrast microscopes, and fluorescence microscopes. Then, there are electron microscopes, which use beams of electrons for incredible detail. The two main types are transmission electron microscope, great for internal structures. Scanning electron microscope, amazing for surface details. Operating procedure of microscope, position microscope in natural light accessible area. Clean lens thoroughly with lens tissue or cloth. Prepare slide, cover specimen, secure slide with stage clips. Adjust focus with coarse adjustment screw. Switch between low and high power lenses for image viewing. Handling of microscopes. Always hold the microscope with both the hands in an upright position while carrying. Don't hold the microscope from the vertical tube and always work on it by standing behind the microscope. Keep the microscope in upright position and do not move the stage in a slanting position. Be careful so that objective lens should not touch the slide in any case. While adjusting, do not move the lens downward, it should always be moved from bottom towards upward direction. Clean the lenses and metal parts including stage with tissue paper or muslin cloth. Place the microscope on the table in the right way. If you enjoyed this video, don't forget to like, share, and subscribe for Biotech Learning.